Not only gift is there, sweet talk is also there. Is that right? Now macrophage is also encouraged, happy. Relationship is advancing. What it will do now? Yeah, wrong things come into your mind. His eyes start shining. What do you think? What macrophage should do? Okay, Gigi will tell us. Yes, what macrophage should do now? His gift has been accepted. Ladylike T cell has also liked the presentation or wrapper of the gift, I must say, and very happy and informed to the headquarter. Then there's some sweet talk, right? That also help in further stimulation. What the macrophage should do now? He will develop a personal contact, physical contact. It's very strange how the molecules in cell interact. Now, this brings one more molecule out. Who knows the name of this molecule? And this will adjust with one more molecule which is already present on the surface of, okay, I'll make it different color so that you don't get confused. What will happen? That a very special contact molecule, it is not released like interleukin 2 was released, but this molecule is not released, and of course this molecule's receptor is also present on the pre-programmed lymphocyte. Now macrophage will express one more molecule which will stimulate another receptor on the T cell, and another message going to the headquarter. Headquarter will get overwhelmed until it start reacting to the situation. I will tell you how it will react. What is this molecule called? B7, that is good. This is called B7 and this is called CD28. I think it was discovered on 28th sequence. So B7 should react with CD28. Now you may be thinking, what's the fun to know all this thing? Let me tell you one thing. They have now the drugs which can block this pathway. They block drugs which block this pathway. Do you think then T lymphocytes will work? No. So drug will be immunosuppressor drug. They suppress the immune response by T cells. And they are planning that if they can neutralize this molecule or block this system, they can control the actions in the immune system in certain diseases. Is that right? Now, this is the primary interaction, and this is called co-stimulation. This was the primary stimulation, isn't it? And this is called co-stimulation. This is antigen and TCR is called first signal. Antigen with TCR is called first signal, and B7 with CD28 is called second signal. To activate the T lymphocyte, we need first signal as well as second signal. Are you understanding? Second signal shows there is ultimate commitment for the action, right? And then T cell become really active. What this T cell will do? It will become very happy that out of so many lymphocytes I have been selected and I have been co-stimulated and I have been specially, you can say, given attention. What this lymphocyte will do? It will activate its nuclear machinery. Let's see what it does. It will activate, suppose gene number 1, I'm putting myself the gene number 1 and gene number 2. Gene number 1 will produce the, of course, messenger RNA and then proteins, and those proteins will go out. These are the protein molecules which are being secreted now by selected lymphocyte. This lymphocyte becomes different than other lymphocyte. It starts secreting special type of molecules. Happy, isn't it? Now, Suhail, you are laughing. Are you? I say this lymphocyte is happy and starts secreting a special molecule. Why it made you so happy? Not happy? Okay, I thought maybe it's special cause of happiness. Okay. Now there's another funny thing. It starts releasing this red molecule. Attention please. This is gene number 1 and here is gene number 2. As gene number 1 is producing the product and secreting the product, gene number 2, it's so interesting thing, start making a protein, and that protein is not secreted, that protein is stuck into membrane of this cell. Is that right? Now you may be thinking what these two genes are doing and their products are doing. Let's see. Then this happy molecule come and fit in here. And as soon as the first molecule stimulates the second molecule give the ultimate happy signal to the nucleus and stimulate a set of genes which are concerned with mitosis. They are concerned with mitosis. And once these genes are stimulated, this selected T cell, which has a TCR against this unique star antigen of that bacteria, the selected T cell start mitosis process. And all other T cells are just standing like this. But the single T cell start, what? Multiplying. It becomes thousands and then millions, right? We say that selected clone has expanded. What do we say? Selected clone has been, yes please, expanded. And this process is called clonal expansion. So this was the process of clonal selection and expansion during immune system activation. So now you see the T cells which were having such TCR which could interact with star antigen, that T cell has multiplied itself, right? So in our body now or in our lymph node, these anti-star T cells are less or more? They are more. So they were clonal selection process and they were clonal expansion. Now look. This molecule which was secreted by the lymphocyte on activation, right? What is the name of this molecule? This molecule is called interleukin 2. This is called interleukin 2. And what should be this molecule called? Interleukin 2 receptor. So when you will talk in a professional way, up to now, let me tell you, whole this thing which I explain, let me narrate now in a very professional way. What we should say, that bacteria entered into your throat, it started multiplying, damaging the tissue, inflammatory response built, blood vessel arterioles dilate, microcirculation endothelium become sticky to the white cells. White cells come into tissue. Neutrophils are first to come, followed by macrophage. Neutrophil phagocytose some bacteria, destroy the bacteria and release their antigens free into fluid, and these free antigens keep on draining through the lymphatic system into draining lymph node. Clear? Meanwhile, macrophages are more professional in handling the antigen. Macrophage initially also phagocytose the bacteria, break down the bacteria into antigens, but they process the antigens with which molecules? Class 2 molecule. Antigens, once they are processed with class 2 molecule, these antigens are expressed